Welcome to everyone. My name is Pietro Roversi and I am a chemistry lecturer at the degree course of Natural Sciences at the University of Leicester. And today I'd like to talk to you about structural biology. I am a structural biologist myself. And what is structural biology? Well, biology, we're all familiar with the concept. It's the study of living organisms. And in the slide, you can see here two typical living cells, a eukaryotic one and a prokaryotic one, not to scale, I have to add. So structural biology is a branch of biology which is interested, therefore, in living organisms, but in particular, it is interested in structure, and this in turn relies on physical chemistry of macromolecules. So structural biology focuses on macromolecules from living cells, and macros in Greek means big, and indeed the molecules that structural biology focuses on are considered rather big. What are they? Well, a class of them is nucleic acids. We may already be familiar with them, DNA and RNA, which have the role of sort of providing information to the cell on how to make proteins, the other a class of macromolecules that structural biology focuses on. When we speak of structure, what do we mean? We mean that we're interested in the position and relative arrangement in space of the atoms that make up the macromolecule, be it a piece of nucleic acid or a protein. Not only that, but because in reality macromolecules move around and are not static objects, we are also sometimes interested in how the atoms of the macromolecules move in space. And you can see here a depiction of how crowded the cell is. And if you think about the fact that the whole of life stems from such random movements in a very crowded environment, you can see the phenomenal difficulty of the problem at hand. So why do we want to do structural biology? Why do we want to know where every atom in a certain macromolecule is and how they move in time? This is because these macromolecules, nucleic acids and proteins, carry out their function thanks to the fact that they have a certain three-dimensional shape and a certain way of moving. So once we know the structure, we can understand how these molecules function. And for example, if we look at this picture, which depicts the spike on top of the severe acute respiratory syndrome coronavirus 2, which is the virus responsible for COVID-19 disease, the shape of this spike is such that it can carry out infection of living cells. So this is an example of what I mean when I say structure is related to function. Of course, once we know a structure and we understand the function, we may hope to interfere with it. And here the metaphor is one that you may be familiar with, which is that if we want to interfere with a macromolecule, we may think of the macromolecule as a lock, and once we know the structure of that macromolecule, we know the shape of the lock, and therefore we can make a drug, which in this metaphor stands for the key that opens or that jams the lock, depending whether we want to stop a macromolecule from working or whether we want to potentiate its activity. So how small are these molecules that we call big? Well, they're very small, and in this picture we go from 100 million nanometers, the size of a tennis ball, all the way down to a tenth of a nanometer, which is sort of the size of a small molecule like water. Structural biology concerns itself with objects that are of dimensions in the range from tens of nanometers, like the antibody in the picture, or a virus, which is like more of a size of 100 nanometers. Now these are very small, and for example, structural biology is not something like scanning electron microscopy, which gives us beautiful pictures of, in this picture we can see yeasts and eukaryotic cells and bacteria. This is still a range of size which is too large for the molecules we're really interested in. Structural biology looks at objects that, as we said, are much smaller. And in this picture, for example, you see a transmission electron microscopy picture where the coronavirus is imaged, and you start appreciating details like the spike that we saw the molecular structure of before, which are those little blue objects sticking out of the surface of the virus. Here we are down to, again, 120 nanometers. But the beauty of such small things is that there are billions of them around. So, and that's where there's an important concept that we will see in a moment. One slide just to say that the fact that they are so small does not mean that they are weak or irrelevant. And sometimes size, like in these infographics, can be used to arbitrarily inflate an object and convey a sort of different concept. In this particular graphic, we can see that viruses and pathogens that throughout history have caused a lot of deaths are represented as bigger if they've killed many more people. So although these viruses are of similar sizes in this picture, the Black Death pathogen is inflated to stand for the more than 200 million people that die of it during the epidemic of bubonic plague in the Middle Ages. The coronavirus, by the way, is down here in the tens of thousands of deaths. Now, structural biology is a branch of biology, but it needs molecular biology and it needs biochemistry. Why is that? That's because if you want to make billions of copies of a macromolecule that you're interested in, you will need to use some cells and persuade them to make many copies of the molecule that you're interested in looking at the structure of. And this is typically done, unless you purify from tissue, which is also a possibility, by molecular biology, and it starts with some DNA cloning, because if you're interested especially in a protein, there will be a gene that codes for it, and we can imagine that this gene is this orange piece of DNA, and people insert the gene coding for the protein of interest into a blue piece of DNA, which is called a vector, forming down here in the corner we see a plasmid. This plasmid can be put into cells, and these cells are called expression systems. They are cells that incorporate the plasmid with the gene of interest and are used as factories for making protein. At the end of this process, you go from the cells that are typically destroyed, and then you need to isolate the protein of interest, or rather the billions copies of the protein of interest, and have a tube which only contains that particular protein and nothing else, other than sometimes a little bit of salt or a pH buffer to stabilize the protein. So with this process of going from DNA coding for a certain protein to an expression systems, you arrive at pure protein in a tube, and that's where the structural biologist will start his or her work. So how is this done? Well, in some sense, because they are so small and they cannot be imaged under an optical microscope, all the various branches of structural biology rely on hitting the macromolecules with something and looking at what comes out of the sample. A little bit, if you want, like hand shadow puppetry, where visible light shines on hands and leaves a pattern on a wall or on a screen, and then you can imagine that based on that pattern that you see on the wall or the screen, you try to understand what the configuration of the hands that gave rise to that pattern, and you can see already that it's not always that trivial. So there are three branches to structural biology, broadly speaking. One is called nuclear magnetic resonance, and what it uses radio waves that are directed onto a sample which is typically in an aqueous solution, and the macromolecules are in the solution, and by collecting the radio waves that come out of the sample, then one can infer the structure of the molecules. The second type is called X-ray crystallography, and before you can get going, you need to grow a crystal from your solution that essentially is an ordered array of the molecule of interest, as small as a grain of salt or sugar, and then you shine X-rays on this crystal. You collect the X-rays that come out on photographic film, or these days on detectors, cameras, and then again you have the structure of the molecule that you hit with the X-rays. The third branch of structural biology has become immensely popular in recent years thanks to the invention of very sensitive detectors, and it is called cryo-electron microscopy. In this case, you are not using electromagnetic radiation hitting your macromolecules, but you are actually hitting them with a beam of electrons, and you record the pattern of scattered electrons. So this is where the availability of large numbers of the molecules comes into play, because you could not conceive of doing NMR X-ray crystallography or electron microscopy with one single molecule, because the signal would be very weak, but you can make them in large amounts. And so for NMR, if you have 200 microliters, which is a fifth of a milliliter of a solution which is 500 micromolar, if you work it out, you'll have 60 million billions molecules in that solution, and that's where the signal comes from, because they are all absorbing and re-emitting radio waves, and that's where you have enough signal to detect them. The same holds for a crystal, which is a very easy way of packing. In this particular example, if it's like 100 microns per side of a cube, of a density around 1 grams per centimeter cube, you would have, for 100 kilodalton protein, something like 70,000 billion molecules in that crystal, and that's very easy to orient in your X-ray beam, and that's where the ease of structure solution comes from having scattering of X-rays from these many molecules. Finally, when you prepare grids for a cryo-electron microscope, you also manage to deposit on the grid hundreds of thousands of molecules, and it's by looking at hundreds of thousands of molecules all scattering electrons that you can reconstruct the shape of each of them. That's assuming that they are all the same shape. So if we come to kind of examples of what structural biology can do for us, and we look in this slide at artwork put together by an artist on the basis of structural biology information on the severe acute respiratory syndrome coronavirus 2, which is the one that gives COVID-19. The structural biology effort that's been ongoing since January earlier this year is aiming at three things. One is to understand the virus. What does it look like? How does it infect cells? How does it propagate? And then, of course, once you know it, you can generate antivirals, i.e. drugs that stop the virus from working. Or ultimately you can produce a vaccine, as a piece of the virus typically that would give rise to an immune response in all the people that are exposed to it, and giving individuals sort of immunity against that piece of the virus. So as an example here of NMR, we can see the NMR ensemble of one of the nucleocapsid proteins from the virus. These are proteins inside the virus shell around which the RNA of the virus is wrapped. And if you look at it, what does it look like? It looks like spaghetti in some places where it's rather mobile, and therefore it means that that protein of the virus does not have a fixed conformation with respect to the rest of the molecule. And in some other places you can see that the 40 copies each in different color that are represented here are all more or less similar. This denotes a rather well-defined part of this protein, and of course this has implication for the function of the protein in wrapping the viral RNA around itself. As example of X-ray crystallography, I want to mention that in the past 4 or 5 months there have been 131 structures of proteins from the virus. And you can see here a panel illustrating the variety of structures that have been determined recently. And I've chosen this one in particular, which represents the structure of an antibody which came from a patient from 2003, a patient of the earlier severe acute respiratory syndrome, not caused by this virus, but from a related virus. So this antibody, which is represented in magenta and cyan at the top of the picture, was isolated from this patient, and researchers in America have recently discovered that that antibody can also bind to a part of the coronavirus given COVID-19, which is the molecule in green. So in here we have an example of how structural biology can elucidate detail of the interaction between our immune system, the cyan and magenta antibody, and the virus, which is in green in this picture. When it comes to cryo-electron microscopy, here I've chosen an example also from the end of March this year. And what we see at the top of the picture, we see in orange a domain from the surface of the coronavirus. And we have on the left-hand side, we have one view, and on the right-hand side, we have another view of a big object. What is this big object? At the top, as I said, there is the domain from the virus surface, and then in the other colors are parts of the human receptor, which is called angiotensin converting enzyme 2. This is on the surface of our epithelia, and what the cryo-electron microscopy experiment captured here is how the virus latches onto this surface receptor before it enters the cells. So if you want to summarize, structural biology enables us to understand.
Washing your hands with soap is possibly the simplest way to flush pathogens down the drain. Soap molecules can interact with pathogens, such as coronavirus, leading to their destruction. Each soap molecule has two parts. The head is hydrophilic, which means it likes to interact with water, while the lipid tail is hydrophobic, which means it wants to get away from water to find similar water-avoiding particles. This is the basis of how soap works. When soap molecules encounter lipid particles on our skin, the tails aggregate around them to form spherical structures called micelles, creating a water-free environment. Similar things happen when they encounter enveloped viruses such as coronavirus. Enveloped viruses carry their genome and supporting proteins inside a lipid bilayer membrane, with the proteins necessary for infection embedded in the membrane. In cells and viruses, these lipids are packed neatly into two sheets with the lipid tails facing inward. The lipid ends of the soap molecules are attracted to the lipids in the membrane. They take advantage of the presence of membrane proteins which can perturb the neatly organized bilayer to insert themselves into the viral membrane. If there is only a small amount of soap, the soap molecules only loosen the membrane. But with more soap, they begin to create micelles around membrane lipids. They are also attracted to hydrophobic amino acids in the membrane proteins, extracting them from the membrane. The structural integrity of a virus is essential for infection. So, washing your hands with soap protects you better from infection than washing with only water, and the longer you lather, the higher your chance of destroying the virus. Washing your hands with...
So, in summary, I hope that you had a glimpse of what structural biology is. And of course, structural biology at Natural Sciences Leicester is an example of an interdisciplinary coming together of chemistry, biology, and physics. And the course is structured in several strands which carry out throughout the course, covering biology, physics, chemistry, and earth sciences. If you want to know more, you can come to the website and in general, you can read about natural sciences course throughout Britain at the website of the Society for Natural Sciences. Thank you for your attention, and hopefully see you in Leicester.